Okay, welcome back everyone to the last in the respiration series. So this is not in the main sequence of respiration, which we have now completed, going through the glycolysis stages, link reaction, Krebs cycle and electron transport chain. Those are your four main stages of aerobic respiration. It is worth noting though that doesn't always happen. Anaerobic respiration is something you should already know that exists, and obviously if you are into your sports science, your physiology, then you'll know a little bit more about it already, hopefully. But we're going to take obviously a biochemical view for A-level standard anaerobic respiration. So it's worth pointing out before we start this what happens where within the cell. This is a recap. So of course glycolysis, the first stage you learnt about, that's in the cytoplasm of the cell. Then it's actually the pyruvate that gets imported into the mitochondria, and everything else takes place within the mitochondria. You can see there the link reaction and the Krebs cycle taking place in the mitochondrial matrix, and then the electron transport chain there being on the membrane itself, moving protons between the intermembrane space and the matrix itself. Okay, so it's worth keeping that in mind as we talk about what we've got to cover today. So how much ATP? So now this is actually a fairly non-trivial question. The process we've described obviously is there to build up a proton gradient, and that proton gradient generates ATP. Originally it was thought that if you looked at an entire mole of glucose, you'd make 38 moles of ATP. So if one molecule of glucose, you'd make 38 molecules of ATP. However, these estimates have been changing through the recent decades. More recent estimates are lower than that, 36, maybe 32. Prokaryotes are actually less efficient than that. We're not going to go into prokaryote respiration, but it's due to the fact they don't really have a lovely environment for this electron transport chain like the mitochondria. They kind of make do with some folded membrane on the outside. So for them it's about 30. That's a bit lower as well. All right, how do we get to these numbers? Okay, why is the estimate changing and so on and so forth? Well, we'll look at one efficiency calculation to start with. This is something you might be asked to do on an exam, and if not, it's just vaguely interesting. So basically, if we fill in this table here, okay, with the amount of things that are on each in each stage, in glycolysis, look there, you've got 2 lots of ATP go in, 4 produced, you get a net output of 2. For Krebs cycle per glucose, you get a net output of 2, right? That's substrate level phosphorylation throughout. Now, when we think about the NADH and the FADH that's produced, now this is where the wiggle room comes in. Each NADH apparently can yield, according to one estimate, 2.5 ATP, and FADH a little bit less, 1.5 ATP. Okay, because it comes in slightly later in the chain, therefore less proton pumping. So the calculation would go something like this. If you top them up, and I'll fill the table in a minute, if you top them up, there are a total of 10 NADHs manufactured, all right, per glucose. Okay, so that's 10 times 2.5, 25 ATP from the NADH. The FADH, right, per glucose, you get 2. Okay, per glucose you get 2, so that's 1.5 times 2. Okay, so 3 ATP from FADH in total from 1 glucose. If you add in the glucoses from the substrate level phosphorylation, the 4, so that's the one from the 2 from glycolysis and the 2 from the Krebs cycle, right? You get 2 plus 2 plus 25 plus 3 plus 32 overall, because as you should have worked out from the last 3 videos, right? We get that many NADs and FADs out of the process up to the point before the electron transport chain. All right. So as I say, it's these numbers here, 2.5 and 1.5, that have a little bit of wiggle room in them. And this energy transfer is not 100% efficient. I mean, no energy transfer is 100% efficient, but there are lots of reasons why this is not 100% efficient in terms of its energy. Okay, now you could well be asked to suggest some of those reasons. If you are, then here are some answers. Okay, now the best sort of estimates are approximately 32% efficiency, which sounds really poor, but is actually much better than most mechanical motors. Right, certainly the internal combustion engine, that kind of stuff, much better than that in terms of producing useful work. But it is actually, 32%, about a third efficient. I've heard a lot of estimates that are even lower than that. Okay, why is this? Well, Reason number one, some protons will leak across the mitochondrial membrane, so they aren't all available to go through the ATP synthase to generate ATP. Okay, I mean, that is because what prevents them going through is, of course, the phospholipid bilayer and the fact that charged hydrogen ions can't make it through. Okay, but if you get enough of them, right, you get a gradient that's strong enough, some will be able to be pushed through. I mean, think like pushing magnets together. If you push hard enough, you're going to get some of them to touch. Okay, so there is some leakage, right, and that is not ideal, but it's what happens. Okay. Some ATP you actually have to invest in moving the bits that you need around. So some ATP is used moving pyruvate into the mitochondria by active transport. And this is one of the reasons why anaerobic respiration starts. If you run out of ATP, you can't actually move the pyruvate into the mitochondria. So you've got to invest some ATP to get the next lot of pyruvate into the mitochondria. Okay. Similarly, you've got to move some other things like hydrogen from reduced NAD made during glycolysis into the mitochondria. There's other things you actively have to move around in the mitochondria, thus using up some of your ATP. So it's not available for other cellular processes. Then, of course, there's the always present answer of heat. It's the standard GCSE physics answer. Just write heat if you're not sure. Some energy is lost as heat. The heat helps to maintain a suitable body temperature for enzyme-controlled reactions. And there is one tissue you might be interested in looking into yourself, which is called brown fat, that has that specific role. There are actually holes in the inner mitochondrial membrane that mean that this gradient just doesn't build up. So ATP isn't being generated, but you're still getting that lost heat. Okay, hibernating animals famously have brown fat. Okay, it's worth looking into if you're interested. So, if we look specifically at anaerobic respiration then, so what happens when you don't have the oxygen, when you switch into an anaerobic mode, the efficiency obviously goes down by far, but you still get some ATP. And all of that ATP is the ATP from glycolysis. Okay, so if you remember, these were the reactions that we learned in the glycolysis video. The glucose gets phosphorylated to glucose phosphate and then splits into two triose phosphates, and it's those two triose phosphates that are turning into the pyruvates. Okay, and that reaction there is what generates our ATP. Okay, so that bit there is really key. What it requires, however, to do that is NAD. Okay, now what we learned from the last video was that NAD is produced in the electron transport chain, where the NADHs are broken down to donate those protons and donate those electrons to that proton gradient. Okay, that energy, and so then the NAD is recycled. If that bit stopped because there's no oxygen to be the terminal electron acceptor, you don't have any of this stuff, the NAD, which means that this bit here can't happen. Okay, so what happens in anaerobic respiration is the pyruvate, right, via another pathway is used to oxidize the NAD, and that looks like that. Okay, so what happens is the pyruvate is reduced itself, so therefore the NAD is oxidized, right? So the reduced NAD is oxidized back to NAD, and you can carry on this conversion of triose phosphate to pyruvate. Okay, and that generates a little bit of ATP, and that's where the ATP from anaerobic respiration comes from. This is obviously not a long-term strategy in you because of what you make. Right, you make lactate or lactic acid, and obviously any of you know about your sports science know that there's only so much lactic acid you can tolerate in your muscles before they cramp up and you stop. Okay, if you are a microbe or a plant, particularly a yeast or fungi and a plant or a plant, you have another step on there and you manufacture ethanol. Okay, you basically chop off one of those carbons, right, to make a 2-carbon compound, ethanol, and CO2, which is still toxic but marginally less so, because these organisms have evolved to live in anaerobic conditions for longer periods of time, whereas you haven't. Okay, so that's why this is happening. The pyruvate can't get imported into the mitochondria, right, because the oxygen's not accepting the electrons from the end of the electron transport chain, so the whole thing backs up in a big traffic jam. More on that in a moment. So the pyruvate is not being imported, so it's just sitting around. We need this NAD back. Okay, we need this NAD back to do this reaction to generate a little bit of ATP rather. Right, so therefore the pyruvate is reduced, the NAD oxidized to recycle this point. Okay, and that is what's going on in anaerobic respiration. Okay, so obviously the link reaction, Krebs cycle, oxidative phosphorylation will have had to have stopped for this situation to happen. Why has that happened? Well, as I just mentioned, Reduced NAD is usually oxidized back to NAD during the electron transport chain. The electron transport chain requires oxygen. Oxygen is the final electron acceptor, okay, in oxidative phosphorylation, okay? But if it's not present, right, i.e. we're in anaerobic conditions, then all of those protein carriers we talk about, right, they stay reduced, okay? They can't get rid of their electrons. So what you end up with, right, is a big traffic jam. So all available NAD and FAD stay as NADH and FADH because all those protein carriers can't accept another electron, okay? So they all stay as NADH and FADH, meaning they can't go back to take the products of the Krebs cycle, for example. So the Krebs cycle grinds to a halt, which then means that the acetyl coenzyme A can't enter the Krebs cycle, and therefore the link reaction grinds to a halt. And if the link reaction grinds to a halt and pyruvate's building up, no more is going to be imported, so that sits around in the cytoplasm, and that's where the previous reactions take place. Okay, and that's what respiration is. It's basically the result. Anaerobic respiration is. It's basically the result of a big old traffic jam. Okay, now this anaerobic path that reoxidizes NADH to NAD is not ideal, right? But at least glycolysis can continue, and at least the cell can produce a little bit more ATP to carry on with those vital processes that it needs to survive. Okay, so that's what anaerobic respiration is. Okay, the formula for animals looks like this: pyruvate plus a bit of reduced NAD gets turned into lactate and oxidized NAD. Okay, if you're a plant or a microbe, right, then pyruvate plus reduced NAD is turned into ethanol and carbon dioxide and oxidized NAD. Okay, and that carbon dioxide obviously bubbles off as a waste gas. Okay, so those two formulae are you're expected to know, and obviously if you were presented this in an A-level question, then you'd be expected to look for what we're talking about. Are we talking about plants? Are we talking about animals? Talking about microbes? Which ones? Okay, now of course this leads to an amazing process that humans have harnessed, which is basically how we have exploited this process of anaerobic respiration in yeast specifically. And of course, that is fermentation and brewing, right? Something that I'm particularly interested in as well, something I learned to do as a student. And if you're going off to university, I recommend you learn this skill as well. It'll make you very popular and save you an awful lot of money. Okay, the idea being if you take yeast and something with sugar in it, if it's the sugar from grapes, you're making wine, the sugar from apples, you're making cider, the sugar from malted grain, you're making beer. Okay, and you bung it in a big bucket like that, right, with your yeast. Your yeast will convert all of those sugars into alcohol, and you'll end up with beer, wine, cider, whatever you're making. Now, just for interest's sake, there's this wonderful little device over here called a hydrometer. Now, what that does there is that basically measures the thickness, right, the density of the liquid. Okay, it's a bit of lead down there, and if it's very, very sweet and sugary, right, glucose makes liquid thick. It will float nice and high, and as the yeast convert that sugar into ethanol via this anaerobic pathway, right, your little measuring thing will sink deeper, and you know when you've fully converted all the sugars into alcohol when it sits at a particular what we call gravity, right, and that's basically the end of your fermentation process. So highly recommended that you learn to do this process, right? It's a very useful skill, and if you can bake a cake, you can manufacture your own alcohol. Okay, obviously not if you're under 18, and obviously it is illegal to sell without a license, but for your own purposes, home brewing can be a useful and very fun biological hobby. And that is the end of the respiration topic. Okay, I hope you've got a clear understanding of respiration now. Do check out some of the other playlists on here for all of your other module needs. I hope to see you again soon. Bye bye.